Hello, welcome. Welcome back to the Headwaters Science Center 3:30 STEM show. I'm Spencer Potter, and today we're going to be talking about photosynthesis. And we're going to be doing a little experiment on some spinach leaves. And I want you guys all to know that this is something that you can do at your house. If you want to experiment with photosynthesis yourself, all the things that you're going to need are some form of vacuum. A simple syringe will do the trick to obtain a strong enough vacuum. But today, for our purposes, we have this very large compressor vacuum that is attached to this type of vessel. And, you know, we didn't have to use something like this, but this is something that we'd like to show off our cool toys here today. And of course, your sample, you don't need a specific brand or anything, just a sample of spinach leaf. I think you can use lettuce leaves and a hole puncher. So the first step that we're going to be going through, I forgot, and some baking soda. Baking soda there's a lot of carbon stored up in baking soda, and we know that plants need carbon to complete photosynthesis. So we created a solution of 100 milliliters of water, tap water, room temperature, and 1 gram of baking soda, and dissolved it into the solution and mixed it up until it was completely dissolved. We are using a chair that's holding on to a specific type of light. We're going to be using that light to help speed up the photosynthesizing process, just so we can see visually what's going on. So, first step, I'm going to get all this stuff going on, and then so we can start recording what we see right away, and then I'm going to talk about kind of what's going on. So the first step is to take your hole puncher and your spinach leaf and punch out 10 circular discs. Okay, and I already did it. I have 10. Oh yeah. So I have I cut them all out. You can see that they're just small little circular sections of the spinach leaf. And I did some ahead of time, so I was all ready and prepared for the show. You may be wondering, why do I have 2 glasses? Well, when we're doing science experiments, we like to observe some different variables, and we're going to be examining light. Light is going to be our variable today. So our constants should be the same concentration of solution in each vial, the same amount of solution, and the same amount of suction pressure should be applied to them. So once you've completed cutting out all your lettuce circles, dump them into the baking soda solution. Okay, and sometimes they just get a little sticky, so having a fork around or something, tweezers may be a good idea. Alright, and do the same for both. Both of these. Okay, once you get your leaf sections into your solution, mix them up. try to get them all so they're not sticking to one another, not sticking to the glass. And then this is where it comes into, we're trying to create a vacuum to suck out all the air that's making these leaves buoyant. You can see, let's make some initial observations. When we put them in to the solution, they were floating. Within the cell, these are plant cells. Within these plant cells, there are little pockets of air. And we're going to try to remove that air and use that key factor, air, as our indicator today. So we're going to try to suck out all the air with our vacuum. You can use a simple syringe, but we're going to use this cool machine right here. So next step would be to put your solution into the vacuum chamber. Yeah. There you go. All right, so we entered the first one into the vacuum chamber. Kink it up. All right, now this might be a little loud. Can you get a good view of the vacuum chamber in there? All right, so I'm gonna turn on the vacuum here. I'm gonna let it go for a few seconds, and I'm gonna make sure that by the time I'm done with the vacuum, that none of these are floating. And it helps a little bit to shake it around a little bit, make sure that they're not sticking to each other and sticking to anything else. And I think that should be okay. All right. So, once you've put your leaf droplets into the vacuum, pour it back into the beaker, and then just take a glance at it, make sure that they don't keep floating. And if they do float, you might need to do it again. Let's see. No, looks like. Oops, sorry. Yep. Looks like I didn't get quite good enough suction because they are still floating up on top. So I'm gonna do it again, and that's okay. Shouldn't be any problems with doing it again. Alright, hopefully this one works. It seems like I can see a little bit of air pockets coming out of the cells. Turn it off. And let's see if this one worked. Alright, now let's make a few observations. Make sure that none of your leaf droplets are floating. Okay. It looks like all of our leaf droplets are sank to the bottom. So let's complete that process again with the beaker number 2. And sometimes these leaf droplet or leaf circles can be a little sticky, so just make sure you get them all in. Alright, we got them all in. So we're going to complete that process again for test number 2, or beaker number 2. Again, we are going for that vacuum, trying to suck out all the gases in the cell, in the vacuoles of the cell, trying to force water into them, make them less dense. Alright. Okay. Let's go ahead and see if our vacuum worked. All right. Let's see what we got. All right. Looks like most of them were able to sync, so I'll just remove that one. Didn't quite fit into ours. Didn't quite get the vacuum that we needed, but so we've removed all the gases from our leaves, and this is going to be a key factor because that is going to be our indicator whether photosynthesis has happened. So place one of your beakers under your light and your other beaker away from the light in a darker area. And we're going to compare and contrast the differences between the light and the dark. I'm going to set a timer here just so I know when to look at them. Okay, so what was actually happening? What are we looking at? Last week we talked about different types of cells, prokaryote, eukaryotic, and then we talked more about eukaryotic cells. Then you divide those into two categories of plant and animal cells. And in the differences between a plant cell and an animal cell were those specific organelles, such as a cell wall and a cell and cytoplasm. And that's where we're going to be looking at. Cytoplasm is the active site of photosynthesis. The chloroplast, sorry, so the chloroplast. And in that chloroplast, there's a structure called a thyroid membrane. And in that thyroid membrane, you can find chlorophyll. And that's really that's like the main factor that drives photosynthesis, because in that chlorophyll is a pigment that is able to absorb a lot of light, and that can be transferred into energy, which can help drive photosynthesis. So photosynthesis, what do we need for photosynthesis? We need carbon and water. So I think the formula is actually I wrote it down. Formula is 6 carbon dioxide, CO2s, plus 6 H2Os. Put that through a reaction, and you'll end up with sugar, C6H12 O6 plus 6O2. So I know that's kind of hard to follow, but the thing that we're that we're really looking at is going to be that the difference between the before and after the reaction is that oxygen is separated, and oxygen is going to be that's a key essential to all living things on Earth. So you and I, we all need oxygen, which we can dedicate and thank our leafy friend here, the lettuce plant, to contributing to the oxygen within our world. So let's take a look at our tests quick. Make sure, again, just make sure that they're not touching anything. We don't have any floating in this light beaker yet. And just make sure that they're not stuck to the bottom of the glass or each other. And let's see, in the dark beaker, looks like nothing is floating yet. So while those are continuing to go through their process to determine whether photosynthesis is happening within these spinach cells.
Wait a minute, Spencer.
Yeah.
Wait a minute. Okay. So you got some pieces of lettuce leaf in baking soda solution under light, and pieces of lettuce leaf, same leaf, in baking soda solution that's covered with not getting any particular light.
Yes.
Okay? So the idea is if those leaves are photosynthesizing,
Yep.
is going on, it's going to produce oxygen. It's going to produce oxygen bubbles. The
Yep.
bubbles are going to be in the leaf. That's going to make the leaf more buoyant, and the leaf sections are going to float to the top.
That is correct.
Okay.
That is correct. And that's what we're looking for. Oh. So my timer at 5 minutes, we'll take a look and see if there's any differences. Again. So is there Alright, well, it's kind of like watching a pot boil. Alright, what do you got, Chuck?
Okay, so you said that you had this formula with so many carbon dioxide and so many waters, and you're going to combine things, and it's going to come out from carbon dioxide and water to an end result. But I remember from a class trying to chart all the stages of the reactions between starting ingredients and the end product. How many stages? Like 26 stages or something like that? There's a happen between the start and the finish?
Yeah, it's not a one-step process. There's many stages in between where you're exchanging ions or electrons and going up and down. And do you remember all those 26 steps?
No, I do not.
I definitely do not remember. Oh, let me draw out the formula here so you can get a visual.
It was incredibly complex. And that happens in every single leaf, in every single little bit of algae, in every little part of a tree. And without that, none of this plant-based material would be around us. It's incredible what photosynthesis does. Without that, we wouldn't be breathing.
So this is what our equation looks like here. So we have 6 carbon dioxide molecules. We have 6 water molecules. H2O goes through photosynthesis, and we're left with a sugar. This is a sugar. That is a sugar, and then 6 oxygen molecules. All right, so our time has passed. Let's take an observation. Let's see. There is 3 different small, or 3 different of the leaf chunks floating just above the bottom. We have one floating all the way on the top.
It's working. working.
So we're getting some sort of gas that must be more buoyant than the water that it's in. It'd be pretty safe to say that we could contribute their buoyancy to their release of oxygen through the process of photosynthesis.
One's all the way at the top now.
Oh, we have 2 at the top
All right.
and 2 hovering below. Let's take a look at our dark beaker. Not looking so good for the photosynthesis. Looks like all remaining are on the bottom.
There they are. They're all done. Ah.
So this is a fun experiment that you can see, witness photosynthesis happening in real time with just common supplies that isn't going to cost you more than, maybe you have to get a syringe. And most people have baking soda lying around their house. So if you want to demonstrate photosynthesis at home, again, all you're going to need is syringe, some form of vacuum, some form of plant leaf. In this case, it was spinach, and beakers, baking soda, a light, and I think that's all I have here, Chuck.
Pretty good.
All right. You want to zoom in here? Maybe we can just watch the photosynthesis happening in real time for a while.
Hey, oh. Hey, there's another one going.
There's another one going.
Pretty cool. Okay, yeah, this could be like watching grass grow, but that's watching photosynthesis too.
All right. Well, so thank you for tuning in for the Headwaters Science Center 3:30 show. If you would like to come experiment with photosynthesis with us in person, come on down. We'd love to have guests and get them hands-on experiments. You're wondering when you can come? That's a great question. You can come. You can visit us anytime, Monday through Saturday, 9:30 to 5. And then stop by after lunch on Sunday in between 1 and 5, and we'll get you set up with a photosynthesis lab.